Welcome to Barry's In the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Well, welcome back, everybody, to In the Interim. I am your host, Scott Barry. Appreciate you joining me here. As on many of these, I have discussed. My wife Tammy listens to all of these and her comments to me about the various podcasts. Also asking, where am I looking in all of this? So you can see I've this sort of setup, and I don't peer into the camera here. I'm looking at my large monitor where I have my notes about the podcast and my other computer down here, and the camera's over here. So I know it's a little bit odd that I'm always looking in the opposite direction. Maybe this is in part my introvertedness of being a statistician. It's hard to peer into the camera. There's nobody there in it. And that's the old joke. How do you tell an introverted statistician from an extroverted statistician? The extrovert looks at somebody else's shoes. All right, so I think it's fair game for me to criticize statisticians, and I may do that again because I am one in this. Years ago, we were involved, Barry Consultants were involved, a number of people, really wonderful effort, in a project called Adapt It. Adapt It was funding from the FDA and the NIH to investigate bringing innovative trial designs into trials that were part of the NET, Neurological Emergency Treatment Trials. And we went in and we took what were going to be 5 different trials. It turned out to be 6 trials we investigated. And what if we looked to innovate the trial design? What was really great about the project is we had behavioral scientists studying that process of bringing innovative trial designs to these particular trials. One of them was, which ended up being ICECAP, cardiac arrest. Patients suffers cardiac arrest. They're brought into the emergency room. All of these particular scenarios centered on the emergency room. And the question is, does hypothermia create better neurological outcomes? There's a period of time where the blood is not flowing in the brain, lack of oxygen, and heat makes that worse. By putting a patient in a hypothermic state, does that create better outcomes? That was just one of 6. Where there was a stroke trial, there was a spinal cord injury trial, there were a number of particular trials. So there were behavioral scientists that were studying bringing innovative trial designs to that. We explored adaptive sample size, we explored response adaptive randomization, we explored futility, interim analyses, multiple arms, modeling, all in the different scenarios. The behavioral scientists in studying this, part of their goal was, what are the barriers to bringing innovative trial designs? Is it the funders? Is it clinicians? Is it regulators? Is it operations? Are it statisticians? What are the various barriers? Interestingly, the number one barrier that they found were statisticians, which is kind of an interesting place, interesting role. And I think it was a different time. This was about 2005 to 2010-ish that we were investigating these trials. It was a different time, but they were hurdles that you had to overcome, that bringing these innovations, the what ifs and all of that. Now, we do this at Barry Consultants. We do a number of what I'll call innovative. And now innovative is self-glossing is better. And I always get concerned saying, we design innovative trials. By some sense, you mean better trials, smarter trials were better. But what I mean by innovative trials is things that are rarely or never been done before. Now, you could argue whether they're worse or better. So I want to leave that aside, but I'll bring that we're doing things that are done relatively rarely or have never been done before within trial designs. I'll call those innovative trial designs. What turns out to be barriers, statisticians in some cases can be barriers. I don't think they're barriers. I think you can present and create scenarios. What are the way that we make it more robust and handle that? What turns out to be a barrier to me, I'll speak for myself, is the scientists who say, I can't define it, I'll know it when I see it. Now, what do I mean by that? The scenario is you're trying to look at adaptive designs, or you're trying to measure, is design A better than design B? You have to be able to understand what's the goal of the trial. What are we trying to do? Are we trying to find the right dose, the right duration? Are we trying to find the right patients? And what is success? If we're trying to compare A to B, we've got to be able to say A does a better job at being successful than B does, or vice versa, or the amount of resources to get the right answer. But you have to be able to define what is good, what's right, what trials would you go to phase 3? And when you want to simulate that and you want to compare that and you want to say, is A better than trial B? If the scientists on the other side say, I can't define what's better, show me the trial result and I'll tell you whether I'll go to phase 3. That doesn't lend itself to analytical comparison. It doesn't lend itself to building a better design, because I don't know what success is. When are we there? What is good and what is bad? That becomes really the enemy to doing innovative designs. Now, I'll talk as we go through here about ways to combat that. I don't think I think you can get out of those individuals good answers in that scenario. So I want to bring up a common scenario where this can happen. And this was, by the way, this was a topic suggested to me as somebody said, oh, I'm sure you did a podcast on this. I'll go find it. And I hadn't, but it's a ubiquitous thing for us. This comes up all the time. And that is, here, I'll do a setup and then I'll get to the topic, is we're sponsors looking to design a phase 2 trial. The phase 2 trial has 2 doses and a placebo, so it's a 3-arm trial. And they're thinking it's a reasonably rare disease. And it is a rare disease. They're thinking of running a trial that is 30, 30, and 30. So 30 patients on placebo, 30 on dose, the low dose, 30 on the high dose. They want to run the phase 2 trial, and then they're going to run the phase 3 trial. And this is a scenario where we know the phase 3 endpoint. There are some scenarios where you're running phase 2 and it's really a wide open space of what the endpoint might be. The endpoint is somewhat dictated by the area measuring clinical benefit. So the endpoint is known. And then they're going to run a phase 3, which is a 1-to-1 comparison of the dose that they select to placebo. And they're thinking that trial might be 90 versus 90, that the effect size that they need to have, they have a pretty good idea of that trial being 90 versus 90, being powered at their effect. So they're looking at a development pathway that is phase 2, which is 30, 30, 30. They get full follow-up on patients. They go through the analysis of that data. They look at that data. They make a decision on going to phase 3. They then submit the protocol. They go through all what I'll call the white space between 2 and 3, that they then run a trial of 90 versus 90 in phase 3. So we said, well, what about a seamless 2-3 trial where you enroll 30, 30, 30. you select a dose from that, and the trial seamlessly moves into phase 3, which is randomization of placebo to the dose that's selected in phase 3. And we can talk about the size of that, but let's for a minute just think about the separate trials was a 90-patient trial followed by 180 patients. The seamless 2-3 trial could be 30, 30, 30. You pick a dose and you enroll 60 against 60. In the second part of the trial, and the patients in the first part and the second part are part of the primary analysis. So the dose that's selected in the seamless trial will have 90 patients against placebo 90, and you'll have 30 on the unselected dose. Now, what does that mean? In the seamless trial, you do have to adjust the alpha at the end if you're going to include those patients because you quote unquote picked the better dose in phase 2. What you cherry picked the better dose in that. And then you are including that data at the end of phase 3. Straightforward ways to adjust alpha and do that. What's great about that and what is kind of amazing is that you have more power, even though it's still 90 patients, than you will have if you run a separate 90 versus 90 phase 3. The power of the seamless 2-3 trial, which is 90 patients followed by 120 patients, 210 total compared to 270 total, it's higher power for demonstrating success at the end of that phase 3. So huge benefits in that scenario for resources. But the other part of it that's just incredible is the lack of white space between them. The time for that trial to read out, the time to then select the dose, start phase 3, enroll another 180 patients. So we simulated that. And by the way, you run into this case of how do we pick the dose between them? And I don't want to spend a ton of time on this. Maybe that's a whole separate podcast of picking that dose. It can be purely on efficacy. It can be a combination of efficacy and safety. It can be efficacy with a DSMB weighing in on whether there's a safety or tolerability issue on it. But for now, let's think about picking the better dose based on efficacy. And in this particular case, the primary endpoint at the end of phase 3 is really a combination of both. It's the clinical outcome of the patient. So we're going to pick one that has the better clinical outcome. That's a case, by the way, that you could run into this enemy that I referred to of, I can't tell you how I'll pick the dose. You need to show me the data and then I'll pick it. But that's not the topic. When we presented this and we showed that this trial of 210 patients has more power than 270 patients, adjusting for alpha, the incredible part of this is they estimated the time between phase 2 and phase 3 will be almost a year. That's just the natural space that will happen if they run a separate 2A and 2B. That space disappears in a seamless 2-3 trial in that setting. We asked them how long that will take. We looked at enrollment. We simulated the trials using the numbers presented. And again, one trial is 270 patients. And the average time to read out of the phase 3 portion of that was approximately 50 months. It was a range to that we had a confidence interval or a prediction interval for the range of that. And the median time for the inferentially seamless trial was 25 months. Just call it 45 months to 25 months. 20 months different, less resources, higher probability of success. Win-win-win in that scenario. The feedback was, no, we don't want to do the inferentially seamless trial. And it's a case where it's sort of, I have this, wait a minute, what do I not understand? This is a very common response, actually, in a scenario like this. Another aspect of this that's so important is the chain of communication. If the communication is somebody said no, and that went through person A and person B, and then we hear the answer is no, it's hard to understand why. And it's hard to ask about it. It's hard to potentially understand, is there a remedy to that no? And many times over the years, I brought this up, that we're trying to do innovative things that are not commonly done. It's not infrequent that somebody says no. Operations says no. Regulators say no. Somebody says no. And understanding why, a lot of times there's a reasonably straightforward way to remedy that, to then get cheaper, faster, stronger. The reason for this, which is a very common reason, is funding. The company is on the smaller side of the pharmaceutical industry. A big pharma may be able to fund the phase 2 and phase 3, and they can set up those criteria and they can run it. What about a smaller biotech? They currently don't have the money to finish the 210 patients. They can run the 90-patient trial. They have the funding for that, but they need to get funding in order to enroll the rest. So that's a very common response, a very real thing. I'm not going to laugh at that in any particular thing, but it's part of designing these trials, innovative trials, is can we combat that? So it's a very common scenario. By the way, it's the importance of being there when somebody says no. It says maybe we have a remedy for that. So can we set up funding solutions and triggers of the phase 2 that triggers funding? And by the way, these funders could be private equity. The funders could be a larger pharmaceutical company, could be a range of individuals that might be interested in investing in the drug. They would like to invest in a drug that's going to be successful in this scenario. So in the separate trials, what typically happens is the phase 2 data reads out and they unblind themselves to that data. And they can share that data with a potential funder, and the funder can play with it. And they go back to this. We know it when we see it. The funder then can decide, will I invest X dollars in this as we move forward for the phase 3 trial? And then I'm a partner with the biotech. If this ends up being successful and the drug makes money, we're invested in that. And we bring in our experts, and we're able to decipher from the phase 2 data whether we're going to be successful in phase 3, what the market share looks like, and is this a good investment for us? And that's a very common scenario, and that's what happens in a typical phase 2 trial. Now, what are we looking to do? We're looking to take that phase 2 trial and setting up objective criteria that would trigger funding. So you go to a funder and you say, okay, you're interested in funding this. Can we define what you would need to see in phase 2 to fund phase 3? If we can do that, we can do that interim analysis where we select the dose, and we can also let the funder know, did you hit your funding criteria? Sometimes this is done separate from dose selection. By the way, this can be done in a phase 3 trial. So this isn't unique to seamless 2-3 trial. That was just the particular recent example that we were working on. Sometimes this happens where the company is ready to run phase 3, but they can't fund phase 3 in its entirety. And a funder says, you know, I'm interested in this drug, but I need to see more. I need more proof of concept of this before I'm willing to invest the big pot of money for phase 3. Sometimes you can start phase 3 and do an interim analysis of whether we hit that proof of concept, proof of funding, that the funder says, yes, if you could jump this hurdle, we would fund the rest of phase 3. If we can objectively set that up, agreement can be made that an analysis takes place during the trial. I'm not going to call it an interim analysis, and I'll come back to that. An analysis takes place. It's conducted by an independent statistical group, an independent CRO, and literally it's a checkbox. Did we hit the funding criteria? Maybe it's 2 or 3 checkboxes. Did we jump the following hurdle? And that is signed by the independent group, and that is given to the sponsor. And we'll come back to who knows and who sees what when. But that checkbox could be presented to the funder in agreement that they've agreed to now fund the rest of the trial based on the deal they have with the biotech. And they know they hit proof of concept in phase 2, even though they don't get to see the data. Now, that's the big question of what you get to see during the course of it. So the data in a phase 3 trial with one of these analyses or a seamless 2-3 trial, it doesn't really, neither one is different. Because as far as regulators and the scientific community are concerned, those patients in the phase 2 part or the earlier part of phase 3, they're phase 3 patients. It is considered bad form, and I'll come back to that, to unblind the data during the course of a phase 3 trial. Sponsors are remained blinded to what is the effect size of the treatment during the course of that. We don't want Jane Doe to be unblinded. We don't want the pharmaceutical company to know that Jane Doe or John Doe was treatment or placebo. Does the data, is there altered? Is there some aspect of knowing that unblinded data that changes the trial? And this is this thing called operational bias. So we'll come back to why typically you don't. But let's just assume that you can't unblind the data, go to a conference, present, ooh, look how good our data are in the middle of the phase 3 trial. Will you help fund the rest? Which is why a lot of times it's a separate trial so that they can do that or present their data, unblind the data. In the middle of a phase 3 with one of these analyses or a 2-3, you can't interrogate the data itself. You don't want to unblind yourself to any individual patient, but you can come back with a limited set of information of, is the data progressing well at this point, above a particular hurdle? Sometimes futility analyses are done in trials a lot. This is different than a futility analysis. What do we call this, by the way? We tend to call it either an administrative analysis or a financial analysis. We'll write into the protocol that an administrative or financial analysis will be done. We don't want to call it futility, because writing in futility means if you don't hit that, we're going to stop enrolling patients. We're going to change the trial. But this analysis doesn't change the trial at all. Doesn't change the sample size, doesn't change how a patient is treated. All it does is trigger funding, potentially. It could be that funder, it doesn't trigger the funding, but the company gets funding another way and the trial continues on. So this is not something that affects patients, the scientific aspect of it. And so we don't want to call it futility. It's a misusing of futility if you do that for funding. Call it administrative, call it financial, and have that done. So let's come back a little bit to why we do that and who knows what when. But if you read the FDA guidance document on adaptive designs, where interim analyses are done, they have a great deal of concern of what they call operational bias. Operational bias it's a hard thing to define. Largely, it's that by knowing the knowledge of the data, it alters the future of the trial or the interpretation of the data in some way. Imagine in the middle of a trial if patients find out that the investigational treatment isn't working very well, and many of them drop out of the trial. Now you've got a huge missing data problem, the interpretation of the trial. You've risked the whole interpretation and the ability to understand is a treatment beneficial. The opposite could happen if there's a really positive signal that patients drop out and go elsewhere, that future patients enrolled look very different, that the investigator will no longer enroll this patient or that patient, or something changes. Now, these are loosely defined, and it's by the way, it's hard to know if you ever have operational bias. It's hard to defend that you didn't. If somebody says, oh, you knew this and it changed your trial, it's hard to prove that it wasn't there. So we do a great deal to make sure that there's no concerns about this mysterious thing called operational bias. So during the course of a phase 3 trial, we don't want the pharmaceutical company, the sponsor, to know the data. We don't want clinical sites to know the data, patients to know the data, and all that. It's important within the scientific credibility of the trial. So in this circumstance, it's not uncommon that you get a piece of paper that says, was this criteria met? And I'll come back to what do those criteria look like? What kind of things do we do for that? But is it met? Yes, no. Is criteria 1 met or criteria 2 met or no? So 3 checkboxes. So what does that mean? Yes, you can reverse engineer from the checkbox. Roughly, are the data above or below this? Largely, that's not considered to create the risk of operational bias. You still want to keep that information relatively confidential. You don't want to publicly disclose, if you can prevent it, that our effect size was above 20% at the interim. Does that cause this potential operational bias? So setting up a relationship with a funder that says you're going to get this checkbox, at the sponsor, maybe somebody in the C-suite gets this as well, a CFO. Somebody who's not going to sites and talking to clinicians and investigators that are enrolling patients. You'd like to create that separation. You'd like the people involved in running the trial to remain as blinded as possible during the course of it. So it might be that the CFO, the CEO sees that data, but otherwise it is firewalled from the rest of the company. And then the funder can get this information, also agreeing not to publicly disclose it, but they agree to write a check if that's there. Now, what kind of criteria do we tend to use here in a phase 3 trial in that? It's not uncommon that we use the predictive probability of trial success. Now, what is that? That is a calculation that's integrating over the uncertainty of the treatment. What's the probability that statistical significance is going to be met when we enroll out the full trial? Sometimes this is called assurance, based on a particular prior distribution in that scenario. Sometimes conditional power is used. Given the effect size we've seen now, suppose that's the real effect size, what's the probability of success? The Bayesian predictive probability of success doesn't assume a single effect. It assumes the uncertainty of that effect given the data we have. It's a very natural Bayesian thing to integrate over the uncertainty of the treatment effect and calculate the probability that we're going to meet statistical significance at the end of the trial. What I've described that is a lot of times, and you'll not be surprised that I'll go to the sports world here. You can watch an NFL football game, you can watch a basketball game, you can go to ESPN, and it will tell you the probability that one team's going to win the game at any point in the game. So at halftime, one team's ahead by 14 points. It might say the probability they win this game is 92%. Or if it's a tie game, might be 50-50 in that setting. That's the predictive probability of success. That's exactly what we're doing in a clinical trial, that we're calculating that, given what we understand now and the strength of the two teams, the strength of the two treatments, what's the probability that we win the trial? Now, a clinical trial is not a game where the high score wins. You've got to win by a point spread. You've got to be statistically significantly better. So if the two treatments are tied at this point, the chance of winning this trial is going to be less than 50%. It depends on what sample size it is in that. If you're seeing exactly the effect size you need to show at the end of the trial, that's where it's about 50-50 that you're going to win the trial. If the data gets a little bit better, you're going to win. If it's a little bit worse, you're going to lose statistical significance. That would be a 50% probability of success. That's pretty good data, by the way, depending on the disease. If this were an Alzheimer's trial, for example, or an acute stroke trial, and you've got a 50-50 shot of winning the trial, you might want to invest in that trial. That's a really good particular gamble in that scenario. Because the outcome is such a positive outcome for you. The revenue to reap in that scenario is positive enough to win the trial. Now, that predictive probability of success sometimes gets confused for power. Power is not something that changes based on the data that you observe. Power is something that if the treatment effect is delta, here's the chance we win the trial. The trial is going to be 80 or 90% power. Every trial is 80 to 90% power. You just figure out what delta makes it powered. So there are times where they say, oh, to invest, we want it to be 90%. That's a really, really high predictive probability. Think of that sports game. So this is exactly the scenario. You're sitting there. You could make a gamble on team A or team B. And at halftime, a team has a 90% chance to win. If you're investing A and you get a double A at the end, of course you're going to invest in that scenario. Now, the people the group that you're investing in understands the odds, and the payout of that is going to be adjusted by it at the time you make that. Now, this is different. You get to make that investment on it. Now, if you say, sure, I'll invest at 90%, it's pretty unlikely. Well, it may be unlikely that the company can hit that early on, but maybe they can hit 50% or 30%. Maybe that's where I would jump in. You can go back to thinking about, suppose I had a separate phase 2 trial. When would I invest? What effect size would I need to see before I'd be willing to invest in phase 3? That can all be incorporated within this administrative financial analysis. And again, the enemy of that is, I can't tell you when I would invest. You have to show me the data. Now, many scenarios, this is kind of a weird concept, this predictive probability of success in the trial. To the people who are so used to building a trial, where they're used to talking about power and type 1 error and all of that, this is all of a sudden a little bit different. They may be familiar with conditional power and they may be familiar with futility analyses. But this predictive probability of success is a little bit of a different sort of beast in the trial. So sometimes they're concerned that funders might not understand that. The interesting thing is funders understand that immediately. That's the world of funding. What do I invest? What do I make? And what's the likelihood that I make it? And that can evaluate that investment. Incredibly straightforward. So this is a powerful thing to funders. Now, why might a funder agree to this objective criteria in it? Well, let's go back to the beginning of this, is If they say, no, run a separate phase 2 and show me the data, and then I might invest, they're investing something that's 45 months to the readout of potential success, and it's a bigger investment. 270 patients is the investment, and it's 45 months as opposed to 25 months and a smaller investment. We get it sooner. It's a win-win-win if we can agree to this potential funding. It's a win for the pharmaceutical company. It's a win for the drug. It's a win for investors. It's a win for patients. It's a win for everybody in this agreement if that can be agreed upon. Now, it comes back to this, I know it when I see it. Can we define when we would invest in that? It might even be a function of, if we hit this higher hurdle, we'll invest, and here's the outcome. If we hit the lower hurdle, maybe they get a higher fraction of the outcome because it's a bigger risk. I don't live in that world, but you can imagine fairly straightforwardly this being a part of this agreement. This is a very common situation. Many groups are in this. Even big pharma needs to get big pharma to agree to it. And maybe large pharma doesn't want to say, we're going to invest in a 1,500 patient trial. We haven't seen enough individually to at the corporate level to say we're going to invest in 1,500. Run a phase 2 trial. Well, turn it into a phase 3 if you're ready for that, or a seamless 2-3 where you have a funding go, no-go internally in the company. That's got to be much easier to do. And again, it's a win-win-win for the pharmaceutical company in this scenario. If you can define it and create it, it's a very straightforward thing to do in the trial. Important part in the innovation part of it. A little bit of a warning here is some of the terminology in this that you'll hear companies talk about a TPP, target product profile. They're going into it where they know to be competitive in the market, they need to see a 20% improvement, a 10% improvement, whatever that 50% improvement, whatever that is, that's what we're targeting for that product. I have to tell you, lots of people talk about that and they all interpret it very differently. Sometimes it's the delta I want to power my trial to win on that. And so if the effect is 30% difference, I want the trial to be 90% powered for that 30% difference. But you could win if you see 20. About 2/3 of that effect, you can win the trial. Some people call the TPP that 20. That when the trial's over, we need to be that value or above to be competitive in the market. Those are two very, very different things. And then there are two very, very different things for funding. So if 30% is what you're powering the trial for, and at the interim you say we need to see 30% or above, it's disconnected with the trial because you can win at 20. Halfway through the trial, if you see 30 and you see 29, that's awesome. You're likely to win the trial. Again, your probability of success is probably hovering over 90% at that particular time point. Now, if somebody says, oh, but you're not meeting TPP, that's the part where the terminology gets in the way of good communication for that. So one of the ways to combat this, I'll know it when I see it, is to show it to them. Show them a data set and say, if we have this data in phase 2, would you fund us? Because it's hard for lots of people, non-statisticians, to think about the quantitative aspects of what is a predictive probability of success, what does this mean, what does that mean? But if I can see the data, if I can see the effect size, that's meaningful to me, clinically meaningful. So we show them a data set and say, would you fund it if we showed you this data set? Sometimes they say, I also need to see this. Okay, we'll show you that as well. Would you fund it? Say, if we see this data, we would fund it. Okay, how about this data set? Would you fund this data set? And we show them example data sets, Kaplan-Meier plots of what data might look like, of estimated hazard ratio, difference in proportions, mean changes. We show them these data, and then you show them thousands. You show them scatter plots of the various effect sizes. And then you say, by the way, this point right here that you would find, the predictive probability of success is 82%, or it's 63%, or it's this. And now here's this other data set, and you say, here's the predictive probability. And they get to understand it, it's meaningful to them. I know it when I see it through that quantity because it's now familiar to me. So while that initially can be thought of as this is the enemy, that person is somebody that you can work with in simulating this, showing this example, showing example data sets, tying it to a particular criteria. Sometimes it changes the criteria. Let's just do the observed effect at that time in that. Many times it can iterate, and a lot of times, by the way, you're making this decision in a seamless 2-3 trial where you don't have complete data on the phase 2 part. That's okay. You might be building longitudinal modeling of this. You might over-enroll phase 2 a little bit to get enough completers because those patients count in phase 3. They're not wasted. Maybe if you're enrolling 1-to-1-to-1 and you over-enroll the three arms, 2 of those arms would continue in phase 3. So if you don't continue the trial and you stop, they've been over-enrolled. But if you continue, only a third of those patients have been over-enrolled. The other ones contribute to the end of the phase 3 analysis. These are all the things that go into these types of trials and these types of comparisons. In order to tackle what is a ubiquitous problem, you have to be able to fund the trial. All right, so I hope you've enjoyed the discussion of funding, jumping hurdles, combating the I'll know it when I see it. And thank you for joining me. Until next time, we'll be here in the interim.